So today's content, we're going to start off with chapter 9 and area study 2 in unit 4, which is talking about population genetics and how that can change over time. So we're going to start off by learning about the gene pool. So essentially, how you should consider a gene pool is that each person counts as one of these circles, right? And each living thing counts towards one of these circles, and these letters are considered their genotypes for a specific specific gene locus, right? So we're going to get into the details of what gene locus are, and what it means to have a genotype like that. But first, here's your learning intention. So today's lesson, we're going to focus on how changing of allele frequencies in a population will have an influence on the gene pool, and how it can be changed due to environmental selection pressures, genetic drift, gene flow, and mutations. So today's lesson, we're going to look at the basics, which is what are all these words even mean? For example, what is mutation and what types of mutation can occur? Also, we will look at what a gene pool is, what alleles are, what allele frequencies are, and genotypes and phenotypes, and then move into the mutation part beyond that. So just a little recap what we covered in the start of the year. What are genes, right? Remember, genes are the coding part of your DNA, meaning that they are the part of the DNA which actually codes for a protein. And only 1.5% of your DNA makes up the coding region, meaning only 1.5% of your DNA is actually responsible for making a protein. So when you have a DNA sequence, basically the gene sequence will be transcribed, and when it's transcribed, it will have a bunch of features within it, right? It'll have exons, it'll have introns, and it'll have to go through another step, which is RNA processing, where it will lose its introns or exons. Introns to keep its exons, and the exons all combine together to code for the protein, right? So I hope you guys remember this, because now we're going to get into a further detailed part of it, which is talking about alleles and how they come about. Okay, so again, the basics. Genes are found in the chromosome. So your DNA is made up of 21 chromosomes, and all your genes can be found on these chromosomes, right? So your genes are made up of 22 autosomal chromosomes and 1 pair of sex chromosomes, and these chromosomes all contain the genes that code for your proteins, and also have all the other region of your DNA which don't necessarily code for proteins. So when you look at your genes, chromosomes, you can see that for every chromosome, there is a copy of itself, meaning that there is a duplicate version of the chromosome that exists, as you can see here, right? So for the first chromosome, there exists an alternate copy as well. So for every chromosome, there exists a duplicate version, which is just a copy of the chromosome. It doesn't necessarily it's the same copy. It's considered an alternate version of the chromosome where there can be differences. And within this chromosome, there are the same genes located. So let's say chromosome 1 has gene X, the same copy that the other chromosome has will also have gene X. But sometimes there can be a alternate version, meaning that it won't necessarily code exactly the same. Both of them will have gene X, but one might code for a dominant trait, while the other codes for a recessive trait, and both of them combine together to determine what your actual phenotype is going to be. So that's when we call it as an allele. So when there's an alternate version of the gene in the other chromosome, we call both of them as alleles. So we're going to get into that in a minute. bit more easier to understand, let me show you the picture down here, right? So, like you can see here, right? Both of these are chromosome, right? Both of them can be chromosome 1 or 2, doesn't matter which chromosome they are, but both of them have the gene B at locus B, and you can see that they both, even though they have the same gene, they don't necessarily code the same type of allele. This one codes for allele B, which is capital, whereas this one codes for the allele B, which is lowercase. What the lowercase and uppercase means is basically, yeah, recessive or dominant. And when they are the same, let's say this one is capital B and the other one is also capital B, we refer to that as homozygous. If they are different, where one codes for the dominant and the other one codes for the recessive, that is referred to as heterozygous. So looking at the examples down here, right? You can see that because these two code different, they are referred to as heterozygous. And because these code for the same capital B and capital B, both are dominant, that is considered homozygous. Okay, so the copies that exist for the same gene, those are called alleles. Okay, so alleles, there's going to be two of them for every gene, because every gene is, sorry, every chromosome has another copy of itself within the DNA. Okay, so each person has two alleles for a specific gene at a specific locus. What does a locus mean? Is the location of the gene region on a chromosome. So if I say uh, individual X has a dominant allele at locus X, you would say that is capital X, because locus X just refers to the name of the gene sometimes, so you don't have to overthink it. If you see it says locus X, that means that you will be labeling it as with the X. You can see that. So when you have a person's individuals, basically what their alleles are going to be for a specific locus. Let's say one person homozygous alleles for locus X, that means they'll have X1 and X1. Another individual might have heterozygous alleles where X2 and X3. And so on. When you start to collect the data of everyone in the population of what their alleles are for a specific locus, specifically locus X, we can collect data of what each person's alleles are going to be. And when we combine it together, form it into this one form, we call that a genotype. Genotype is the genetic, where's that? Yeah, genotype is the genetic composition of an organism at a particular gene locus. So if I say at locus X, the genotype that organism X, organism A has, you would look at something like this, right? So for example, all these different organism sheep have different genotypes at locus B. So this one has a genotype where it's both are recessive alleles. This one has a genotype where both are dominant alleles. This one has a genotype where one is dominant and one is recessive. So that's how you should be referring it to. Okay, the genotype is the genetic makeup. The B refers to the locus, which is the location within the chromosome, the region where that gene is going to be found. And the letters indicate what type of expression is it. Is it dominant expression or is it going to be recessive expression? And whether it's homozygous is determined by whether both are the same alleles or whether they are different alleles. If they are different, we call that as heterozygous. If they are the same, we refer to that as homozygous. Okay, so this initial part of like trying to understand what each of these words mean is actually very important, because beyond this, you'll be building up the knowledge of these words and you'll be asked questions based on that. All right. So like I said, once you start collecting information about a particular locus within a population. Let's say I want to see what everyone's genotype is at locus X in this room, right? Then I'll start collecting data about this. Let's say for me at locus X, it's a homozygous where both are X1. Okay. And then let's say for, I don't want to use names because it's recording, but let's say for individual number 2, when I collect at locus X, I'll see that one of their alleles is X2 and the other allele is X3, and their genotype is written as such. So genotypes will always come in pairs because there's 2 copies, right? So this one, they've labeled it as X1 and X2 or X3. Sometimes it can be as just capital B and lowercase b. Those are the 2 ways that they can write genotypes usually. So don't be confused if you see this kind of version or the other version of the sheet. They're just writing down the genotype, and you need to be able to identify that it's just different alleles labeled as X1, X2, or X3. This one doesn't indicate whether it's dominant or recessive. It's just referring to the different alleles that can exist, and it can show you whether they are homozygous or heterozygous. Okay, so once you've collected the information about the genotypes for a specific locus, which is locus X, we have it like this. That's when we calculate something known as the allele frequency. Basically, like I said, right, each of these are alleles. There's an X2 allele and an X3 allele, and there are 3 different alleles that can exist. How do we find out what is the allele frequency is basically very simple maths. We calculate how many X1s there are. Let's say we count all the X1s, and we find out there's 12 X1s in the gene pool. And then we calculate how many alleles are there in total, right? That's 60 of them. So to find out the allele frequency of X1, we just take the total number of X1s divided by the total number of alleles in the gene pool. And that's how you find out what the allele frequency for that specific allele is. All right? Who isn't understanding this so far? Anyone need a re-explaining? If you do, please let me know because this is kind of important. Okay, make sure you understand what an allele is. Alleles are the individual alternate versions that a gene can have, and they can be either the same to make it homozygous, or they can be different to make it heterozygous. Right? And within a living thing, there will always exist 2 alleles for any locus because there are 2 copies for every chromosome for living things. All right, moving on. So another example, we have total number of alleles in the population. There are 32. How can we find that very easily? Is just count how many organisms there are times 2, because each organism will have 2 alleles. So 16 organisms times 2 is 32. That's why we can find 32. And now let's find out the frequency of allele B in the capital form. So we just count how many capital B's we can find. There's a total of 12. So the 12 divided 32, we get a 37.5%. So 37.5% of all the alleles found in this population are capital B, okay, or dominant B. If you look at frequency of allele lowercase b, you can see that there's a total of 20 lowercase b's found in the found as alleles, and when you divide it with 32, which is the total number of alleles, you get 62.5. So that's how you should look at it. So that's what a genotype is, right? Genotypes are both the alleles combined. That is referred to as genotype. That is the genetic composition of an organism at a particular locus. So it will always have two alleles present for the genotype. For the phenotype, that's basically the expression of that genotype. So by having a homozygous dominant genotype, what is the expression that will come about? So for example, one example of a phenotype is your eye color, right? Your genes code for your eye color, so that means that is your phenotype. Another example is basically your hair color. That is also a phenotype. Your genes, your genotype for your hair codes for a characteristic, and that is a result of the expression, gene expression and the environment. So that is a phenotype. Lastly, genetic diversity is referred to the variation in genetic makeup of or alleles within a population. So, like we saw in the picture with the sheep just now, in a population there can exist a lot of variation between the alleles, right? Some can be homozygous dominant, some can be heterozygous, and some can be homozygous recessive, and so on. There can exist a lot of variation within a population. That can even be the case right now in this room, right? If we sample a specific locus for any trait, you will see that there is variation that exists. Some of us could be homozygous dominant, that would look like this, where both are capital. Some could be heterozygous, where one's capital and one's lowercase. And some could be homozygous recessive, where both are lowercase b. And each of those gives a different expression. So something like eye color, there's going to be variation in this room for that specific locus that controls that phenotype. So everyone understanding so far? Okay, moving on. So alleles naturally want to stay unchanged. That is their natural state. They want to stay as it is since fertilization all the way to death. But there are natural phenomena that can cause it to change. This is what we refer to as mutations. Mutations are basically permanent changes to a DNA sequence, and the causes for that can be referred to as mutagens. So mutagens, we refer to that as the cause of a mutation, and there can be a lot of mutagens. Basically, anything that can cause a mutation is referred to as a mutagen, where stuff like UV radiation, atomic bombs, uranium ore, mustard gas. There are so many mutagens that exist in the world, and even something like cigarettes can be considered as a mutagen because they do cause rapid mutation. They do cause mutations within your DNA, so they are considered as mutagens. That's basically anything that can cause a mutation to occur inside your body. And why is it important for mutations to even happen? Mutations are considered. Why is it important? Is because any changes within that 2% of coding regions can actually be very detrimental. Because remember, that 2% of your DNA that codes for proteins requires it to be precise so that it can make sure the protein is made correctly and that protein will fold correctly. So what controls the protein folding is all the different amino acids that make it up. If even a single amino acid is changed, then the way the protein is folding will also change. And sometimes that can even lead to the protein becoming non-functional or not being able to emerge altogether. Sometimes that also can be non-harmful, such that the mutation doesn't do anything. But there are different types of mutations that can occur. We will look at the different mutations in a bit. But that's why mutations can be kind of dangerous, is because if those mutations go target the specific 2% of your coding regions in your DNA, it can lead to changes in the expression of that particular gene, which can alter the folding and functionality of the resulting protein. So now the next question is, can these mutations be passed on to the next generation? So when you have kids, would you pass on any mutations you accrue throughout your lifetime? And that depends on basically where the mutation occurs. So throughout your body, most of your cells are non-reproductive cells, right? We call them somatic cells. Okay, somatic cells are basically every other cell in your body that doesn't act as a reproductive cell. Basically, something that's going to be passed on to the next generation. Anything that's going to be passed on to the next generation, your reproductive cell, we call that as germline cells. So make sure you guys are remembering those two words. Okay. Yeah, here. Okay. So germline cells, we consider those cells. Somatic cells has basically every other cell in your body. So for males, germline cells would be sperm. For females, that would be your ovum. Basically, any mutations that accrue within those germline cells will be passed on because you're basically using those cells to reproduce. So those cells that accrue the mutation, those germline cells that accrue the mutation will actually be passed on to the next generation. Whereas any other cell in your body, the somatic cells, they're not going to be passed on to the next generation. So any mutation that happens at somatic cells will not be passed on and will not be considered as being heritable. Right? So make sure you guys can always use the words germline and somatic because they will use those words instead of reproductive cells or any other cells. So you need to be able to understand what a somatic cell is and what a germline cell is. And why is it important for mutations to exist? So sure, they can be dangerous, but they also have a purpose for humans. They also have a purpose for living things, which is that it creates and introduces new alleles in a population. So remember how I said that within a population, there exists a lot of variability in terms of alleles. Some of you might have homozygous dominant, some of you might have heterozygous, and some might have homozygous recessive. So mutations can also act to increase the genetic diversity by changing the alleles and creating new alleles that didn't exist in a population before. Basically, if an entire population is homozygous dominant for a specific locus, sometimes mutations can change that to suddenly introduce homozygous recessive or heterozygous genotypes for that specific trait. That is why it can be necessary to have mutations as well. But it's a balancing act. Too much mutation can actually be detrimental to the point of killing you. And if you don't have enough, it might not give you enough variability. So sometimes it can be good, sometimes it can be bad. Usually it depends on what type of mutation it is going to cause. Okay, so when we think about mutations, since they're happening by chance and there's no control of where or how it's going to happen, there can be three versions that can happen in terms of mutation. One is a deleterious mutation. This is basically where the mutated allele is suddenly producing a protein that is going to have a negative impact on the individual. Either that or the protein is not going to be functional anymore. So deleterious can be the protein has have a negative impact on the individual fitness. Fitness basically means survivability. So this basically means that the protein has become non-functional or the protein can't emerge at all. So that is what we call as deleterious. Another one is advantageous. This is where the mutated allele suddenly improves the survivability of the organism. So somehow rather, that mutation is producing a protein that actually makes the individual survive better, right? Maybe it's producing more of a protein, or maybe it is producing that protein in a different version that can act as a advantage for the individual. Another one is a neutral mutation, where the mutated allele still produces the intended protein without any change. Where this can actually happen due to the way genes are coded. Sometimes one change in the gene doesn't necessarily change the protein itself. It can actually end up coding for the same protein nonetheless. But yeah, each of these can happen depending on how the mutation occurred and what type of mutation occurred, right? So essentially, yeah, like I said, enhanced functioning protein can be considered as advantageous. Normal functioning protein produced can be considered as neutral. And if it is abnormally functioning protein, basically not doing what it's supposed to do it or losing its function altogether, that is considered as deleterious mutation. These are the three versions that mutations can take form as. There are different types of mutation that can cause these three things to occur. We're going to also look at that in a bit. But like I said, in just now, right? Mutation can be a balancing act. Some mutations are so dangerous that can be basically fatal to you in just a few minutes, whereas some mutation you can survive for your whole life and not even notice it. So, one example of a mutagen that is very minimal is solar radiation, solar UV. In fact, you get solar UV every time you go out and you don't even notice it, but it does cause mutations on your skin. In fact, if you ever get sunburn, what's happened is that that UV radiation has been so extreme that that region of your skin, the DNA has damaged to a point it can't function anymore. So it's basically dying off that part of your skin. That's why it's so sensitive and it feels so painful. Basically, that region, all the DNA on that skin has been overtly damaged and can't function anymore. So the rate of mutation for normal solar UV is quite low, but if you get it overexposed or over concentrated over a sustained period, it can be leading to cancer. Another one is smoking. Smoking has the cigarettes have chemicals within them that can actually act as a mutagen as well, such as benzene and formaldehyde. Okay, and that can actually lead to mutations happening within your lungs, and continued exposure can increase your risk of cancer. And my personal favorite, which is the last one, this is a case that happened in Chernobyl. If anyone watched the Chernobyl show on HBO, this is a specific roof at Chernobyl where there was so much radioactive material that just spending more than 30 seconds to a minute can reduce your life expectancy to just 1 or 2 months. So it's so high. The radiation levels there are so high. The rate of mutation will be so fatal that your lifespan would literally be reduced weeks, if not months. So mutations are actually generally dangerous depending on how extreme they can be. Because they are random, they are unpredictable, and sure, sometimes they can be advantageous, but a lot of the times they can also be deleterious, and that is why they can be potentially dangerous as well. Right? So, like I said, there's two types. There's a heritable kind of mutation, which is considered as germline mutations, and then there's somatic mutations, which only stick to the specific organism. That's the one that doesn't get passed on, right? Specifically, remember how we looked at CRISPR and how we can modify a living organism. And if we modify the living organism right after fertilization, that living thing will be able to emerge with that edited genome throughout its life. So if we can do that, then any mutations that happen naturally to germline cells will also do the same thing. So any mutations done to germline cells can potentially be copied multiple times during the embryonic stage, where let's say the mutation happened to the egg cell before fertilization. Since that egg cell has the mutation, after fertilization, every time the one cell reproduces to become an embryo, it will carry that mutation along, and it will make sure that mutation is spread throughout the living thing. Whereas if a mutation happens to the somatic cell, which is after the full organism has emerged, that mutation will specifically be affecting that one region. So think of it like this: if you get a sunburn, there's no chance of you passing that sunburn on to your kids because it's happening at one region of your somatic cells. But if you, let's say, get some kind of mutation happening to your reproductive cells, like your sperm or your ovum, there is a chance that mutation gets passed on and your kids will have that mutation emerge throughout their entire body. So, getting some. So that is how you should think about a germline mutation versus a somatic mutation. So any mutation that happens to somatic cells will only cluster around the cells that were exposed to the mutagen, and that won't necessarily spread through all the cells in your body. Whereas any mutation that happens to a germline cell before fertilization can potentially be passed on to every single cell that emerges from that, and then every cell that emerges from the living thing itself. All right, any questions? So remember how I said that mutations can change the allele? So you can see it here, right? So in this population, everyone is homozygous recessive. Everything has a recessive copy, a recessive allele for the the locus A, where both of them carry the allele for the recessive trait, and then suddenly due to mutation, the recessive allele has now become a dominant allele, and now there will be suddenly an emergence of a heterozygous genotype within the population. Even though originally there were no heterozygous, due to mutations changing the alleles, there is suddenly a heterozygous genotype that emerges. So, are you guys following me so far, right? You know what a genotype is, you know what alleles are, you know what a phenotype is, you know what a gene pool is. A gene pool is basically all the genotypes within a population. Allele frequency is the number of that specific allele within that within that gene pool, and mutations can change the alleles present within a population randomly based on the type of mutation that occurs. All right, moving on. So when you think about mutations, there's the types of mutations. We've looked at, there's three ways mutations can manifest, right? Deleterious, advantageous, and neutral. Those are the forms that mutations can take. What are the different types of mutations? There's specifically two categories that we can split them up into. One is referred to as point mutations. The other one is referred to as block mutations. And as the name refers to, one is talking about a specific point in your DNA that's going to be changed. The other one is talking about a specific block in your DNA that's going to be changed. So there is also one more type that we'll look at later on, but these two are the most common ones that you need to be able to identify. So when we think about point mutations, like I said, it involves a single nucleotide base. So remember, your DNA is a nucleic acid. What makes up a nucleic acid is multiple nucleotides that have bound together to become a polymer known as nucleic acid. So when we think about a point mutation, this is where one specific nucleotide in that nucleic acid polymer is being mutated, and there can be a few ways that can happen. One is called a silent missense mutation, a nonsense mutation, or what can happen is that a nucleotide is suddenly added in or deleted out. So there are four ways it can happen, and they're also categorized differently based on the addition or deletion of a nucleotide. It will cause something known as a frame shift mutation. We'll look into this in a bit. The other one, block mutations. This is when the mutation involves a larger cluster of nucleotide bases. So instead of just one nucleotide that's being mutated, it's a whole region of nucleotides that will be affected. And there's a bunch of ways that can happen. One is known as duplication, deletion, inversion, translocation, and insertion. Right? So we're going to look into each of them. So hang in there, right? So first one, point mutation. Remember, this is one where it involves a single nucleotide base being changed, and that can happen in three ways. One is that it's either being substituted, meaning that it's being changed with another nucleotide, or it's going to be deleted, or another nucleotide is going to be suddenly added in. So three ways it can happen. The original nucleotide can be changed with another nucleotide, or suddenly that nucleotide in question can be deleted out of the entire sequence, or suddenly a new nucleotide is just added in to that sequence. and each of them will affect it in a different way. So let's look at it. So the first one, the first type of mutation is known as a silent mutation. Why is it referred to as silent mutation? Is basically it does not change the protein that's going to be made. So you can see it here, right? Right. So in this is your original. This is your original copy of the sequence at DNA level. When we make it into mRNA, remember that DNA, when you transcribe it into mRNA, this is your template strand, this is your coding strand. So T will code for an A, T will code for an A, C will code for a G. Okay, remember that? And when you make it into a protein, AAG, if you look at your table for the proteins, it will code for a protein amino acid known as lysine. Okay, that is the normal expression. So let's look at what a silent mutation does. So a silent mutation, a silent point mutation will change one of the bases, one of the nucleotide bases, and it will suddenly become a T. Okay, why is this referred to as silent? Is because it does not affect the final protein that's going to be made. Because remember, in the table for the protein production, there is repetitive. Okay, I suddenly need to bring that up to show you guys what I mean. Just give me a sec. Right, so no, you need to go anywhere. Just show me that. So you can see here, right? So one protein, one amino acid can be coded for using multiple codons, right? And so why is it that the codons are so close together? Is because they are basically accounting for point mutations. Sometimes a point mutation can be silent because let's say the A has been changed to a G, it will still code for lysine. So that's why it's referred to as a silent mutation, because the end result, the final amino acid that's produced, is still the same. So it's referred to as a silent mutation. And this is the mutation expression where it's a neutral expression because no changes have been made. Sorry, no advantageous mutation was made. Neither was a deleterious mutation made. It's a neutral mutation because nothing has changed essentially due to that mutation. That is why it's called as silent. The next one is a nonsense mutation. This is when that one change in the nucleotide sequence has suddenly caused it to become a stop codon. Remember what a stop codon is? It is the codon that codes for a stop within the transcription, right? Translation, right? When the polymerase DNA polymerase, when the RNA polymerase DNA, when the polymerase is reading, why am I saying this? When the tRNA is reading the rehash my brain. When the ribosome is reading the codons and is coding for it using the tRNA, when it reads a stop codon, it will terminate the translation, ending the amino acid sequence. That is what a stop codon is. So if the mutation that occurs is such that it suddenly becomes a stop codon, it will basically terminate the translation of that mRNA sequence, and that can be very detrimental. Imagine this is literally the second codon of the sequence. It's suddenly mutated into a stop codon, so the protein won't ever be made, right? That's why this can be a deleterious mutation. More often than not, a nonsense mutation is going to be deleterious because it stops the expression of that gene when it's not supposed to be stopped. And then next is the missense mutation. This is basically when it codes for a different amino acid due to the change in the codon, right? So let's say it's supposed to be supposed to be T in the middle, it was suddenly point mutated to become a C, and TCC, when changed into mRNA, it's AGG, and AGG codes for, I don't know what this amino acid is, ARG, instead of lysome. So that is a whole different amino acid. And like I said, within a protein sequence, even one change in the amino acid can potentially be deleterious because it will impact on how the protein is able to fold. And if the protein can't fold appropriately, that means it won't be able to be functional altogether, right? So there are two types of missense mutations that can occur. One is referred to as conservative, and the other one is non-conservative. You guys don't really need to know this yet. If we do, I'll get to it in a later chapter. For now, I just need you guys to remember the different types of point mutation, right? There's silent, where there's no effect in the amino acid sequence. There's missense, where the change in the nucleotide codes for a different amino acid altogether, causing a different folding structure at times. There's nonsense mutation, where suddenly it codes for a stop codon, and then there's a frame shift mutation, which is basically when it does this down here, right? So let's have a look at this picture down here. So frame shift is caused by two things that can happen. One is an addition of a nucleotide, and the other is a deletion of a nucleotide. So let's look at it in terms of an addition. So this is the normal sequence, where this is the DNA, this is the mRNA made from it, and then this is the amino acids that it produces, right? So it's supposed to produce FAIR, PAIR, glue, and glue. That is the normal sequence. So everything you can see here for the first two codons are the same, nothing's changed. And then at the third codon, specifically the first nucleotide in the first third codon, you can see that something has been deleted or no, added. So you see it's only supposed to have one G, but another G has been added as a nucleotide. And what does this happen? It literally pushes all the nucleotides further down the line. Suddenly the codons are read much differently. So all you see, right, just because one more G was added, look at how the codons have changed. Basically, they are misread every single time beyond that point, because codons are 3 by 3. As soon as you add one in, that order is changed, where it's all pushed one down the line. Do you see that? Who doesn't get this? If you don't, please raise your hand, and I'll try to use another way. All right, think about this, right? If I got everyone in this class to stand in a line, and I said we're going to do 2 by 2, and then suddenly I join someone in the line, doesn't that push everyone's partner, change the order? The same way, that's what's happening here. Imagine these nucleotides are standing in a line, and I split them up 3 by 3 by 3, and I said, however we are arranged is how you're going to be grouped up. But then I suddenly join someone in randomly, that pushes everyone one step further back, right? So by adding one nucleotide in, the reading of the codons will change such that this A will be pushed back to the next codon, and this G will be pushed into the next codon, and so on and so on. And this will keep happening until the very end of the mRNA sequence, and even the stop codon will be shifted such that it's read differently. So it might not even stop, and it might just keep going on, right? So that's why a frame shift mutation is that dangerous, because more often than not, that will cause the entire sequence to be misread altogether, right? Because one addition changes the way the codons are going to be read, and after that has happened, there is no telling how many amino acids are going to be coded wrongly. As you can see here, right? Just because the codons change, the two amino acids that are produced subsequently are already wrong, and probably every other amino acid that comes out is going to be wrong. And that's for addition, and same thing will happen for deletion. If addition pushes it further by one, deletion will push it back further by one. So look at here. So we start off with per, and then where the A is supposed to be, there has been a deletion, leaving the T to take that position. pushing everything further back by one. So where the C is supposed to be, suddenly the C is in the second codon. And where this C is supposed to be, it will move into the third codon, and so on. Basically, all the codons move back by one, all the nucleotides move back by one spot, and then the codons are all looked at read differently. Anyone confused about this? Okay, it's just think this is going to be an easier thing if I demonstrate it, but maybe we'll do that at the end of the class. Okay, so that is the 4 different types of point mutations that can occur. Remember that a point mutation is when a single nucleotide is changed, and there can be 4 versions of that mutation: silent, missense, nonsense, and frameshift. And like I said, each of these mutations can be take a form of either being neutral, deleterious, or advantageous. More often than not, a nonsense mutation is going to be deleterious because it's coding for a stop codon where it's not supposed to be. So that is going to be deleterious more often than not. Or a missense mutation, sometimes that can be advantageous as well. Basically, by changing that one amino acid, maybe it can make the protein better at its function. There is no telling what type of missense mutation that might be. There's so many versions that can occur that some can actually be advantageous as well. And a frameshift mutation, more often than not, is going to be deleterious because it's not just changing one amino acid, it's changing every amino acid that's going to emerge after that. So that's going to completely change the folding of the protein. And when that happens, the protein becomes non-functional, or it basically won't even emerge. All right. I think bringing water next time. What like we talked about point mutations, the 4 different versions that can take up, right? So this is the normal codon as you see it. UUA, frameshift mutation is when one codon is either added in or taken out. This one's showing one that's added in, and because one is suddenly added in, the codon has suddenly changed from UUA to UUG, and the next codon will start with an A instead of what it was supposed to be starting with, right? So this is for an addition. Deletion would take away the A, bringing back the nucleotide from the next codon into this codon, causing it to be a different codon as well. A silent mutation, that's when it's a point mutation, something has been substituted, but instead of it changing the final amino acid that's being produced, it's still producing the same amino acid, basically making sure that no changes can occur. No changes occur to the protein and the folding later on. A missense mutation, that is when it codes for a different amino acid suddenly, and that can either be advantageous or deleterious depending on the change that has occurred. And a nonsense mutation is when it codes for a stop codon instead of a normal amino acid, and that is more often going to be deleterious because it's stopping the expression of that, it's stopping the translation of that mRNA into a protein where it's not supposed to happen. So imagine this, I am making a cake. No, not making a cake. So imagine the protein translation is at like 30%, and suddenly it read a nonsense mutation. It found a nonsense mutation causing the translation to suddenly stop. So the protein is only 30% complete, meaning that it won't emerge properly, meaning that it is going to be deleterious. So a shift in the reading frame causes the issue because all the intended codons will end up being read wrongly. Basically, anything beyond that frameshift mutation will all be read wrongly because there's a change in the reading frames of the codons. Should be on Teams, yes. Oh, yeah. This is very heavy on you guys. You don't have to know all these diseases. I'm just putting this as a reference where you can see all the different diseases and problems that can emerge due to something as simple as one nucleotide changing in your coding regions. For example, cystic fibrosis, sickle cell anemia, Huntington disease, Tay-Sachs disease, and I can't pronounce that one. But basically, all these are happening due to a point mutation happening at a specific gene within a chromosome. For example, let's look at one of them. Let's focus on sickle cell anemia. Who knows what a sickle cell anemia is? Anyone heard of it? Yeah. What do you know about it? Yeah. So this is what your normal blood cell looks like, which looks more like a donut with no hole in the middle. It just has a dip in the middle. Well, a sickle cell looks more like a C, right? The blood cell suddenly looks like a C. And this is all due to the fact of one point mutation happening at the HBB gene on your 11th chromosome, where the substitution of the adenine into thymine at the 6th codon position leads to the replacement of that amino acid with this amino acid, and that in turn causes every protein that emerges from that gene to have this mutation and emerge with a amino red blood cell that looks like a sickle cell. That's the whole. We're gonna get into inbreeding as well. Don't worry about that. Inbreeding is not as simple as, oh, you're related, you have sex. Yeah, you're related, you reproduce, and you're gonna immediately emerge with a disease. There's more complicated. It is more complicated than that as well, because there is a reason why you have a higher chance of passing on diseases when you are inbreeding. That we'll look at that in a bit. Not a bit, in the next chapter, I think. Right? So now that we've looked at point mutations, which are mutations happening in a specific part of your cell, let me just make sure I know how many slides are left. All right, we're almost done. Good. Right? So block mutations. This is a larger section of your DNA. So a larger section of nucleotides are being mutated. And this is significantly more dangerous because it can basically change an entire region of your nucleotides. And this is usually happening at the level of chromosomes, right? So this can happen in a few ways. One is as a deletion, which is a removal of an entire section of DNA from your sequence. Another one is duplication. This is when an entire sequence of your DNA is duplicated again. Another one is inversion, where basically the sequence of the sequence of your DNA has been altered to be inverted, right? Instead of going A, B, C, D, E, F, it's suddenly inverted to be A, E, D, C, B, F. So it's been messed around all around. And then last is translocation, which is basically when between two chromosomes, they have switched places altogether, right? So one is all capital, the other one's all lowercase, and translocation causes them to switch places with each other, right? So look at the EF, which is capital, moved into this chromosome, and look at the lowercase, which is moved into that chromosome. Okay. Like I said, block mutations are happening at a very large section of your DNA, and usually this is happening at the level of chromosomes, where entire sections of chromosomes can be changed, right? So here's an example of deletion. So this is the normal chromosome. The deletion block mutation will basically cut off that certain part and delete it off from the chromosome, suddenly changing the entire sequence of your chromosome. Duplication is basically when an entire region of your chromosome has been duplicated all over again. So if you see the pattern, it's exactly repeated again. Inversion is basically when it is split upside down from the normal order, and translocation is when two chromosomes have basically switched sections of their DNA with each other. So let's say the yellow chromosome, in this sense, has replaced replaced with the gray chromosome, and the gray chromosome has replaced with the yellow chromosome. Okay, any explanation required for this one? So this one, like I said, depending on how it's expressed, can either be advantageous, deleterious, but more often than not, it's going to be deleterious or advantageous. It's not hardly ever going to be neutral because this is happening to a very large section of your DNA, sorry, large section of your chromosome. So hardly would it be neutral. It will always either be advantageous or deleterious altogether. So can any other forms of mutations occur? There are larger scale mutations where instead of block mutations, it changes entire chromosomes altogether, not just sections of your chromosome, but the entire chromosome altogether is mutated. And that is when there's two versions of that. One is called aneuploidy, and the other one is called polyploidy. So what is one and the other? Aneuploidy is basically a chromosomal abnormality in which an organism suddenly possesses an incorrect number of total chromosomes. So instead of having 22 autosomal chromosomes and one pair of sex chromosomes, they suddenly have maybe 23. Sorry, we'll look at examples of one and the other in a bit. So this is caused by the addition or loss of an individual chromosome, where a human being is supposed to have 43 chromosomes, right? Including the two sex chromosomes and the 22 autosomal chromosomes. That's a total of 46 chromosomes. If the total number of chromosomes is lesser, like 45 or then 47, that is called as aneuploidy. Okay. Polyploidy is basically when a chromosomal abnormality in which an organism has a different number of chromosomes for each set. So remember, there are a total of 23 sets of chromosomes within a living thing, and 23 of those have two copies of each chromosome. So that's why there's a total of 46. Polyploidy is basically when instead of having two copies of each chromosome, they can either have one or three copies that can change the entire number of chromosomes within the organism. So this is talking about the total number of chromosomes within the organism being changed, whether it's increasing or decreasing. This is basically talking about whether the number of chromosomes per set is different. If they have suddenly 3 chromosomes for each set instead of 2, or whether they have 1 instead of 3. So for human beings, I've told you, right? All of us have 2 copies of each chromosome. That's why we are referred to as a diploid organism. Okay, when there's only 1 copy of a chromosome, that is called haploid. When it's 2 copies of chromosome, that is called diploid. Okay, so this is an example of aneuploidy, right? So this one's an example of aneuploidy. This is a normal human chromosomal karyotype. We call this a karyotype, by the way. This is basically your chromosomes aligned based on the number that they represent. So this is the first chromosome, second chromosome. Like I said, each chromosome has a copy of itself. So that's why there's 2 per chromosome. And then there's a pair of sex chromosomes per living thing. And females have 2 X chromosomes as their sex chromosomes, and males have a X and a Y chromosome. That is the normal karyotype. In a aneuploidy karyotype, you can see that all of them have the same number of chromosome sets, and the only thing that's changed is the total number of chromosomes, where one of the sets suddenly has a extra chromosome, right? This one, instead of having 2, it suddenly had 3, changing the total number of chromosomes from 46 to 47. That is aneuploidy. Polyploidy is the number of chromosomes in the sets all together have been changed, where each set suddenly has 3 chromosomes instead of just having 2, and this can greatly impact the survivability of a living thing. All right. So I can give you an example of aneuploidy causing a disease or causing a developmental problem is Down syndrome. Down syndrome is actually caused by the 21st chromosome having a extra chromosome, right? So that's a form of aneuploidy because only in the 21st chromosome there's an extra chromosome suddenly. Instead of having two, there's three, and that in turn can be linked to Down syndrome. Okay, I don't really have an example of polyploidy because, like I said, if this happens in a human, the survivability is reduced significantly because there's just too many problems that emerge from it. For aneuploidy, the survivability is impacted, but there is a chance that they can still have a life beyond that. All right. So, as trying to condense all of it into a summary, gene pool. This is basically the collection of all the genes, all the genotypes and alleles within a specific population and can be represented using allele frequencies as well. Genetic diversity, this is looking at the different alleles that exist within a population and how there exists variation between different organisms. Some of you might have homozygous dominant, some of you might have heterozygous, and some might have homozygous recessive. That is a good thing. Having that variation is a good thing because we're going to look at why it's important in the next topic. But basically, it's very important that genetic diversity exists within a population for a lot of reasons. Mutation is when a specific change in a permanent change in the DNA occurs due to environmental factors or possibly mistakes in copying the DNA as well. And it can happen in a few forms. One is a point mutation, which also includes the frame shift, by the way, and also block mutations. So point mutations is a change in one nucleotide where it's either being substituted, added, or deleted. Block mutations is when an entire region of the chromosome is being changed. And then there's your aneuploidy and polyploidy, where the total number of chromosomes, the chromosomes themselves are being mutated to either have a different number of chromosomes or a different number of chromosomes per set in the karyotype. What can be causing these mutations is referred to as mutagens. These are basically anything that can introduce mutations to DNA, for example, like your UV or smoking or different mutagens. And why can this be a good thing? Is because in a population where there's no genetic variability, no genetic diversity, mutations can potentially introduce variation to allow for genetic diversity to exist. All good? Okay. Heavy stuff, huh? All right, let's look at this, right? In a mutation in the eighth nucleotide in the sequence was changed from a C to a G. So in these kind of questions, your first move should always be underline which nucleotide they're going to be they're talking to you about, right? So a mutation in the eighth nucleotide in the sequence. What is the eighth nucleotide here? Right? So it was changed from a C to a G. So essentially, anytime you have this kind of question, always underline, highlight, whatever, label that nucleotide in question, because this is also a point of careless mistakes where people see it correctly for the first time, and then after that, when they're looking at the table and looking back, they look at the different codon or different nucleotide. That's going to just cause you to read it wrongly, right? So let's break it down, right? So what is that? What is the codon that nucleotide is a part of? ACC, right? ACC, and what is it changed to? ATC. All right. Another thing to note here: the following nucleotide sequence occurs on the template strand of the DNA molecule. So, what is the mRNA molecule going to look like? Right? T. Is that going to be a T? Yeah, everyone needs to rehash their genetics component. Remember, T cannot exist in your mRNA sequence. What is it replaced to? Sorry? If this is your DNA sequence, right? ATC, your template strand, what is your mRNA sequence going to look like? UA. G. Okay, and what does UAG code for? So the original codon, what would it have code for? UGG. Right? And now UAG is coding for a stop codon. So what's the answer? You can't read the answers. Oops. Where's the mouse? Alright. Who thinks the answer is A? Raise your hands. Who thinks the answer is B? Raise your hands. Who thinks the answer is C? Raise your hands. Who thinks the answer is D? Raise your hands. Right? A? Actually, not because I thought when it stopped, nothing changed. But like, but originally what it would have been, yeah. So essentially the mutation has changed the amino acid sequence from a normal sequence that would have continued on with more and more amino acids into a shortened version. So the answer is A, because a nonsense mutation will stop the mutation in its tracks. Sorry, stop the translation in its tracks. And when translation is stopped at a point where it's not supposed to, the peptide chain that is coming out of it is going to be suddenly shortened more than it's supposed to be, right? So that's why the answer is A.